Okay, with an understanding of DNA and genes, genes being the information stored in DNA, we're ready to understand animal development. So animals, multicelled organisms, animals and plants, all derive from the single fertilized egg. The process can be diagrammed like this. We've got a sperm going to meet the egg. If this is human, this will have 46 chromosomes. And then the cell goes round and around the cell's cycle. During interphase, it duplicates its chromosomes and then enters mitosis. In mitosis, the duplicated chromosomes get delivered into daughter cells. Now, in reality, the cells stick together, forming an embryo. Here's a picture of maybe an 8-cell or 16-cell embryo. But here we're showing the cells separated so we can see the sort of ancestry. At the right side here, then, what we would have is we would have all the cells of the adult body, or the baby to begin with, but then it'll grow up into an adult. And notice we've identified this one up here as a skin cell, a nerve cell, blood cell, muscle cell, liver cell, intestine, etc. Down here we would have the sex cells produced in the sex organs. But the point is that if you look at an animal, it is composed of specialized cells. So these cells perform different functions. So we're going to call them specialized cells. The cells back early in the embryo do not look like any of the cells that will make up the animal later on. So something's happening here. The cells are specializing. Cells are becoming different types of cells that do different jobs, and then they all cooperate, right, to keep the animal alive. So one of the puzzles of development is to figure out how is it that cells specialize. Let's take a look at some of the specialized cells as they might be in the body. Well, we know muscle cells, they generate a contracting force in our body to move our bodies. And inside muscle cells are two very important proteins called actin and myosin. These are rod-like proteins, and the myosin, can't see it here, but they have little barbs that pull on the actin proteins. And so when those little barbs flex, the whole muscle cell shortens, and that generates a force at either end of the muscle cell. So that's what moves our bodies. Over here we see a scene in the lung. Here is one of the air sacs of the lung. So when we inhale, air comes in. Air has oxygen. Oxygen diffuses through the wall of the lung, and then through the wall of the blood vessel, and then into the red blood cell. So the red blood cells are filled with a protein called hemoglobin. Very important protein. It's what carries oxygen. So the blood cells will travel back to the heart, and the heart will pump the blood around to the body, and the oxygen that is carried by hemoglobin will be delivered to all the cells of our body. Then the blood comes back to the heart, is pumped through the lungs again. It has picked up some carbon dioxide from cells in the body as they burn sugar. The carbon dioxide diffuses into the air sac. We exhale the carbon dioxide, inhale a new batch of air. But notice we've got different types of cells cooperating to perform this function. We've got lung cells making up this hollow chamber. We've got capillary cells making the walls of the blood vessel, and we've got red blood cells carrying the oxygen. So back to our puzzle then was, how can cells specialize into different types of cells? Now the puzzle's even more pronounced because let's take a look at what each of these cells have. They all have chromosomes. Now you'll recall what a chromosome is. It's a long molecule of DNA, and it's often wrapped around proteins. We don't see the proteins in this picture. Here we just see a long molecule of DNA, the double helix. And each chromosome then has the recipes for multiple genes, right? So each gene is a recipe for a protein. And so we can imagine a chromosome is just a long molecule of DNA, and each sequence here would be a gene, a different gene. Now it turns out there's a lot of DNA that doesn't code for any proteins, but that's another story. We want to think of a chromosome as consisting of a long molecule of DNA with multiple recipes for proteins, so we would say multiple genes. But here's the puzzle then. We know that mitosis duplicates chromosomes, so the daughter cells have identical sets of chromosomes. Yet somehow cells with identical sets of chromosomes are becoming different. Now, one way to approach a solution to the puzzle, how can cells with the same chromosomes become different, is to use an analogy. Let's think of the chromosome as like a book, and the different genes would be pages in the book. Now, if we imagine then the 46 chromosomes as a set of books, each chromosome with multiple pages, genes, then the idea is that while each cell has the full library of books, maybe they don't use the same recipes. So it would be like we all have access to the library, but people going into the library read different books, and so they leave with different information, different knowledge. In the same way, all the cells have all the 46 chromosomes, but they may be using different portions of the chromosomes. In other words, expressing different genes, and so they become different types of cells. So if we think about our two types of cells, the red blood cell and the muscle cell, imagine here's an immature cell, and it is destined to specialize into a red blood cell. It does so by turning on the hemoglobin gene. The hemoglobin gene, when it's turned on, the DNA unwinds and messenger RNA is made, and the cell makes the hemoglobin protein. For the muscle cells, the cell is going to turn on the actin and myosin genes. Making those proteins, the cell will specialize into a muscle cell. If we were to peer in the nuclei of these cells, let's take a look at what's happening then to a portion of the chromosome, a little stretch of DNA. So let's first start with the red blood cell. So imagine we're looking at some of the DNA in a red blood cell. Here's just one portion of a chromosome in a red blood cell, or the immature cell that will turn into a red blood cell. Notice what we have here, the hemoglobin gene. So remember, a gene is just a stretch of DNA. So this sequence of letters of DNA specifies how to put together the amino acids to make the protein, to make the hemoglobin protein in this case. Notice we have a messenger RNA is being constructed. So these letters are complementary to the DNA letters. And that messenger RNA will detach, go out into the cytoplasm, and then that temporary copy of the recipe will be used to make the protein. But the point is, if we take a look over here to the left, we see that this bit of DNA here is the myosin gene, and this bit of DNA is the actin gene. Notice they are both wrapped up around those packaging proteins. Now, we saw the packaging proteins when we thought about how does a chromosome condense during condensing stage, and we said, well, the DNA, that long molecule DNA, wraps up around the packaging proteins. But here we see another type of function for these packaging proteins. They are, in a sense, used to help turn genes off. So whenever a gene is wrapped up, it is off. The cell does not have access to that recipe or these recipes. So this is what's happening in a red blood cell then. The hemoglobin gene is turned on, meaning the DNA is separated, messenger RNA being made, the cell will make that protein, but the actin and myosin gene is turned off. And if you can imagine then what's going on in the muscle cell, it would be sort of the reverse, right? The hemoglobin gene is turned off, and the actin and myosin genes are turned on. So when we think about how cells specialize, we want to think about each cell has the full library of chromosomes, right? So here we have 1 to 23. And then since you get one from each parent, we really have 23 pairs, or a total of 46. You only see 23 here. And we can think about each chromosome as a long molecule of DNA with multiple genes. So in this diagram, the idea is wherever you see an M, that's where a muscle gene is turned on. So those are genes that are turned on in muscle cells. Wherever you see a B, those are genes turned on in blood cells. And maybe there are some genes that are turned on in both types of cells. But you can see then a different pattern of gene activity across the chromosomes. On chromosome number 1, there's a gene up here, a muscle gene turned on in muscle cells, but it's not turned on in blood cells. But down here we have a gene on chromosome 1 that is turned on in blood cells, but not muscle cells, and so on across the 46 total chromosomes. So we can think of cell specialization as cells activating different sets of genes in the library that are the chromosomes. Here's another diagram then. Let these little bars represent the library of chromosomes. So again, from an original cell here, a parent cell, two daughter cells can begin to specialize in different ways if they are activating different genes. So a different pattern of gene activity will lead these cells to specialize in different ways. The blood cells will produce proteins needed for blood cells, and the muscle cells will produce proteins needed for muscle cells.